Hello students, this is Professor Johnson, and in this video we are going to talk about photosynthesis. Specifically, we are going to look at how plants capture light energy, use that light energy via the light-dependent reactions to create ATP and NADPH, and then how the NADPH and the ATP are then used by the light-independent reactions of the Calvin cycle to fix carbon into organic molecules such as glucose. Finally, we're going to look at the process of photorespiration and talk about how plants deal with this process. So plants capture energy using organic pigments. So pigments are just molecules that have specific colors associated with them. So they end up absorbing some wavelengths of light and reflecting others. Plants reflect green light, so they will absorb light mainly in the red and blue wavelengths. Now, this is due to the presence of chlorophyll, the main pigment in plants. There's two types of it, chlorophyll A and chlorophyll B, that create a spectrum that helps increase the wavelengths that are absorbed by the plant. The plants also have carotenoids and other accessory pigments that help broaden the wavelengths that are captured. But the main ones are chlorophyll A and B, and they absorb wavelengths that the plant then use to excite electrons and use that to capture the solar energy and put it into the chemical bonds of organic molecules. All right, so if we look at how this light energy that's captured is used, we need to talk about the light-dependent reactions. These are reactions that occur in the thylakoid membrane. We have two photosystems. These are the spots where the light energy is actually absorbed. So we have photosystem 1 that occurs in sort of the later steps, and photosystem 2 that occurs in the earlier steps. They're named after when they were identified, so their name can be a bit confusing that photosystem 2 comes before photosystem 1. So what's happening? So first off, a photon of light comes in. The photosystem 2 is going to capture this light energy, and this light energy is going to bounce from from one molecule to the next in the photosystem until it ends up in a chlorophyll molecule of the reaction center. Once it's in the reaction center, it's going to excite an electron. So that energy gets passed on to the electron. The electron is going to be excited, and it's going to leave the photosystem 2 and pass through a series of proteins that are going to act much like the electron transport chain acted in cellular respiration. So the electrons have high energy. As they go through this, they lose some of that energy, and that energy is used to pump protons across the thylakoid membrane, so that you have a high concentration of H plus within the thylakoid lumen and a low concentration of H plus out in the stroma. Now, this concentration gradient can be used to create ATP via our old friend ATP synthase. This is the same enzyme that we saw in the electron transport chain of the mitochondria. So the protons float down their concentration gradient, and as they do, they're going to take ADP and an inorganic phosphate, combine them together, and you will get ATP. So very similar to cellular respiration, you have a proton gradient that runs ATP synthase that synthesizes ATP. Now, the electron that was excited in photosystem 2 needs to be replaced, and it gets replaced with an electron from water. So the electron comes from water, and because water is losing an electron, that means that water is going to be oxidized, and oxidizing water will result in producing oxygen. So we have now produced oxygen because we took an electron away from the water molecule, and that replaces the electron that the photosystem 2 lost. So now let's go over to photosystem 1. It too gets excited by a photon of light. That photon of light, the energy that's in it, gets passed from one molecule to the next until it ends up in a chlorophyll molecule of its reaction center. This is going to excite an electron, and it's going to be passed off to a protein that will take NADP plus and convert it into NADPH. So NADPH is accepting electrons, so it's being reduced in this process. Now, photosystem 1 needs to replace the electron just like photosystem 2 did. However, instead of using water, it's going to use the electron that photosystem 2 passed through the electron transport chain and replace it with that once it's lost all of its energy. So you replace the electron from photosystem 1 with the electron that ultimately started off in photosystem 2. So at the end of the light-dependent reactions, we've produced ATP via a proton gradient and ATP synthase. We've reduced NADP plus to NADPH, so we have an electron carrier here. And we've produced water, or we produced oxygen from water because we took an electron from water to replace the electron that was originally lost due to the excitement from the photon. All right, so this brings us to the Kelvin cycle. So the Kelvin cycle is going to take the ATP and the NADPH from the light dependent reactions and use them to fix carbon. Fix carbon means to take carbon dioxide and synthesize it into organic molecules such as glucose. So it requires ATP, or it requires energy, and ATP provides the energy for this process to occur, and it involves reducing the carbon dioxide to the glucose molecule. And so NADPH is going to provide the electrons for doing that process. It's called a cycle because you ultimately start and end with RuBP. This is a molecule that's going to be incorporating the carbon dioxide, but before the end of this process, we're going to reform the RuBP so we end up back where we started with it. Ultimately, the enzyme that's responsible for this process is known as Rubisco, and there's something that Rubisco does that is going to actually limit the efficiency of this process. And so what it can cause is something known as photorespiration. So Rubisco, in addition to taking carbon dioxide and fixing it so that you can form glucose, can take oxygen and fix it and incorporate it so that you're actually releasing carbon dioxide instead of capturing it. So this is going to reduce the overall efficiency of cellular respiration. Now, this will occur when the stoma of the leaves, so the stoma is certain pores that are found in the leaves, are closed. They close when it's hot out because it'll want to conserve water within the leaves. So when it gets hot out, the stoma close. This is going to trap gases within the leaf. So as photosynthesis occurs, you're going to start producing more and more oxygen and using up the carbon dioxide. So now you're going to have higher amounts of oxygen within the leaf and lower amounts of carbon dioxide. And if you have higher amounts of oxygen, it's going to be more likely to be used by the Rubisco enzyme and go through this photorespiration process. Then carbon dioxide will be used to form glucose via photosynthesis. So overall, photorespiration is going to limit the efficiency of the photosynthetic process. So how do plants deal with this? Well, there are three different strategies. So we have the C3 plants, which deal with it just kind of like by ignoring the problem. They live in climates where photorespiration isn't severely limiting. And so they'll just end up saying, this is cost of doing business. I'm just going to take the carbon dioxide directly into the cell, incorporate it into the Kelvin cycle, and capture it to make glucose molecule. So that's just the Calvin cycle as we've seen it thus far. Two other types of plants are going to do something instead to minimize photorespiration. So we have C4 plants, such as the maize over here, that are going to apply a spatial strategy. So they have two different cells. They have one cell where they actually are going to capture the carbon dioxide. So the carbon dioxide comes in, it gets captured by a 3-carbon molecule to make a 4-carbon molecule, and then in a different cell of the plant, so they're going to take this 4-carbon molecule, put it into this other cell of the plant. It's going to release the carbon that was captured and incorporate into the Calvin cycle in that cell, and then take these 3 carbons back to the other cell so that it can go through this process again. So it's spatial solution. It ends up capturing carbon dioxide in one cell and fixing the carbon dioxide in another cell. All right, so that was C4 plants. CAM plants, on the other hand, are going to make use of a temporal solution. So it's based on time. They do their carbon capture at night. So carbon dioxide comes in at night when it's cool, the pores are open. They will capture it into a 4-carbon molecule. And then during the day, when it's hot out, they close their stoma pores so that the water can't evaporate when it's hot out. And then they're going to release this carbon to be used by the Calvin cycle during the day. The Calvin cycle can run because we now have the ATP and the NADPH produced from the light-dependent reactions to keep the Calvin cycle going. So they capture carbon at night, they fix it during the day, and so that helps minimize the process of photorespiration. All right, I hope this was informative, and I will see you guys in class.